As part of our work in trying to identify which are the epigenetic drivers of lymphomagenesis, we did a screen for the dependency of diffuse large B-cell lymphoma cells on the sirtuin class of enzymes. There are 7 human sirtuin proteins that have different kinds of enzymatic activity. And by performing a screen for their dependency in a panel of lymphoma cell lines, we found that the one sirtuin that seemed to have the most heavy dependency in DLBCL, to our surprise, is a sirtuin that is localized only in the mitochondria and is not actually in the nucleus. And so this is SIRT3. SIRT3 is a protein deacetylase. Not all the sirtuins are deacetylases, but this one is a protein deacetylase. It's in the mitochondria exclusively and is linked to metabolic programming as opposed to epigenetic programming. And surprisingly, this need for SIRT3 seems to be throughout all lymphoma subtypes. There's no distinction based on the genetics or expression profiles. And the reason for that is because lymphomas are cells that are very highly dependent on generation of metabolic precursors to maintain their very rapid proliferation rate. Lymphomas are one of the fastest and most extensively dividing of all tumors. The fraction of cells that are dividing lymphomas are among the highest of all tumors. And that means they need a lot of fuel. Normally, in the germinal center reaction, the germinal center B cells from which lymphomas arise have only a limited capacity to fuel. They are able to generate metabolic precursors, building blocks that enable them to undergo proliferation. But once they start proliferating, they cannot maintain that pace of building new precursors to keep on dividing, and so they have limited replicative potential. What we've discovered occurs in the case of lymphoma is that at some point, this protein SIRT3 becomes highly overexpressed. It's not mutated, but it becomes highly overexpressed. And what it does is to make B cells now able to be fully self-sufficient in terms of generating constant amounts of precursors. Why? Because by deacetylating mitochondrial proteins that are involved in the Krebs cycle, that makes them very efficient at using amino acids as a source of building blocks. So amino acids that come in from serum or whatever now can be efficiently utilized by the Krebs cycle to generate all the metabolic precursors needed to make nucleotides and lipid and other proteins. And if you, in fact, stop SIRT2 from acting, the cells basically will starve to death. They will, in fact, eat themselves in a process called autophagy. And that autophagy kills these lymphoma cells because they just have such a pressure to replicate that they have to keep on eating their own components until they die. So to see if this could be harnessed as a therapy, we worked with a sirtuin biochemist from Cornell University named Henning Lin, and we're able to develop a compound that is able to inhibit the class 1 sirtuins and to localize specifically to the mitochondria, and therefore only selectively hit SIRT3, but not the other class 1 sirtuins, SIRT1 and SIRT2. And these compounds were able to recapitulate all the effects of the genetic loss of SIRT3 and had very powerful anti-lymphoma activity both in vitro and in vivo, causing them to undergo this autophagic cell death process. And so we're excited about that and working now to develop our clinical lead compound so it can be translated in the future for use in patients.